大家好，我是谢德荣博士我说中文说的不好，所以今天我用英文。So I'm Dr. Michael. So right now, last week I have a flu, so my voice is a little bit cracking. So I hope that you can understand what I'm trying to say. So today I'm going to talk about the genetic engineering E. coli. For what is the connection between the small little bacteria and the green solutions for the sustainable biochemistry. Press here, right? So my research field, as I just mentioned, I'm working with the genetic engineering. So I use genetic engineering and modify the genetic material inside the bacteria, and then to create the strain that is desirable for any production of any kind of biomaterial. Like, for example, right now I'm focusing on the bioplastic, PHA, PLA. So you can see it right now, what I'm holding right now is a petroleum-based plastic bottle. And this one, although it's very thin, it will take a few hundred years to be completely disappear from the environment, which is opposite to what I'm holding right now is my sample of PHB. So this PHB, if you put it into, like, let's say, marine environment, plus a little bit of the UV light, then something like, for example, if this tube is made of PHB, then it can be completely disappear in less than a year under the marine environment. So for today, I'm going to talk about the 12 principle of the green chemistry. So, and what does the E. coli do with these 12 principles? And then I'm going to introduce to you the basic of the genetic engineering. And also I'm going to share about the case study of my most recent study based on the bacteria strain named PHB8. So you can already see that the name PHB, that is for production of PHB. So first, I would like to give my appreciation to Dr. Keller, who has already given a very detailed explanation regarding to this 12 principle of green chemistry. So I think now I just need to tell what is the relationship of the E. coli and these 12 principles of green chemistry. So for the first one, pollution prevention. So as we know that it's better to not produce any pollution than cleaning it up. And for the E. coli here, we actually don't clean up anything. We don't prevent anything. We don't even need to prevent the pollution. We use the waste. We use the waste directly to produce the product that we want. For example, in my case, the PHB production in my recent study, I use crude glycerol. So crude glycerol is a nasty waste from the biodiesel production. So let's say that if we produce 1 ton of biodiesel, then we're going to produce 10% of that, the same amount, which is a crude glycerol. And the crude glycerol is actually very toxic, then not many living organisms can use that. So the second one, for the atom economy. So just make it simple. You have 1 banana, and you want to make a smoothie. And so 1 banana is equal to 0.9 smoothie plus 0.1 banana waste. So it's just like that. In the E. coli, in my case, I use E. coli, and E. coli eat the waste, feedstock, to produce the product. And my job is to trying to modify, to optimize the genetic material of the E. coli so they can consume, they can convert as many of this waste feedstock as possible to become something, to become the valuable product. And for the hazardous prevention, actually I think a lot of people here is a student from Professor Chu, right? So I believe that many people, they also have some experience in working with fermentation. And let me tell you, doing fermentation, we don't care about if something is flammable or something is too toxic. If you smell it, if you inhale, or if you touch your skin, it doesn't harm you, anything from the fermentation. It's the same stuff with the safer chemical. So you see, when I'm trying to make the fermentation, what I need to add into the fermenter is inorganic salt, low concentration. And maybe just add something like a sodium hydroxide or chloric acid, very low concentration. So all of the chemical used in that fermentation of the E. coli is safe. And also solvent. We don't use any specific solvent. All I need is some water to dissolve the salt so that E. coli can eat them. And for the energy efficiency, so you see that bacteria, they're very small. You cannot even see them, so they use very less energy. It's unlike when you're raising the pig or raising the cow or something like that. Then you need to give them the space to live. You need to make them happy. You need to make sure that they are warm or they are cool. So, but for the bacteria, we just give them the very little space for them to live. Give them food, and they grow up and give us a product. For the number 7, renewable feedstocks. So as I just said, we use the crude glycerol. That is actually, we can say that is renewable. Or if you don't want to use some waste feedstock like that, we always can use something that we can grow. So right now, like to produce the POA, the nature work, they have the whole farm of sugar cane. They make sugar cane and they use that sugar to feed their bacteria so they can produce lactic acid as the precursor for the bioplastic PLA, polylactic acid. And for the derivatives reduction, so we know that the less step, the better. So all of the reaction in the fermentation using bacteria like E. coli, many reactions just happen inside that single cell of the E. coli. All we need to do is just to give them the food so they can live and then make the reaction to produce our product. So as you can see that if we use the chemical, any chemical reaction, we need to give them a certain conditions so they can happen. And that is just for one chemical. How about we have 10 reactions? 10 reactions, we have 10 set of conditions. We need to separate into maybe 10 reactors. But for the E. coli, all the bioreaction just happen inside the E. coli. And we just need only one reactor. And for the catalysis E. coli, they make the catalysis by themselves, which is called the enzyme. And designed for degradation. So as I just tell you, PHB on my hand, 1 year under the sea with UV light. And for the RT monitoring, so we just need to have a very simple control for the reactor. And of course, because from the beginning, when you culture the E. coli, is the possibility to have any hazardous incident is already very low. Okay, so that is the relationship between the E. coli and the 12 principles of the green chemistry I just briefly introduced to you. And now, go to the basic genetic engineering. So This all start with the molecule called enzyme. So you see that if you want the reaction to happen, you need to give them the activation energy. And for the case of E. coli, we use the enzyme. The enzyme is made of protein. And if you can control the protein synthetic, then you can control the biochemical reaction to produce any bio-based product. But where does the enzyme comes from? So I think I would like to have some water. Excuse me. So now I'm going to talk about how the enzyme is synthesized inside the E. coli. So everything starts with the process of translation. So we have translation here. The protein is synthesized by the ribosome inside the bacteria using the mRNA. So I believe that a lot of people here, you heard about the mRNA from the vaccine COVID-19. And with the amino acid in the reaction called the translation, and we have the protein. And the mRNA is synthesized by the RNA polymerase based on the very code of life, which is a DNA. So DNA is made of 4 molecular nucleotides. The short name is ATGC: adenine, thymine, guanine, and cytosine. So that ATGC are just the same, people by people, species by species. They are all the same, ATGC. So this code, yeah, they're all the same. So all the genetic designing works are based on that molecule, which is a DNA. So I would like to introduce to you, this is a simplified PHP production process inside the bacteria. So we can already see that we have 4 chemicals, right? And 3 reactions. And each of these 3 reactions required 1 specific gene to happen. And here, let's say that this is a kind of this DNA sequence of the gene. It's not 100% match, but I just give you an example. But you can see that it's very long. So like this far A, it has 1,182 base pair. And this one, far C, is even 1,770 base pair. So the question is, how can we work with so many texts like this? So we use bioinformatics. So for example, this is the vector NTI software. So you can see that all of the gene right here, the position of the gene, the order of the gene, like far C first, and then far A, and then far B. And the detailed sequence of the gene is all written here, like in this software. So to understand this map is very important for the genetic engineer. So we can do the planning, designing, all of the strategy that we wants to optimize our production. And we also can predict what is going to happen in our production. So this is the simplified scheme for all of the genetic works and also the fermentation. So first, we get the interest DNA that we want from the source that we think is reliable. And we do some techniques like we do PCR, we use a restriction enzyme cut, and then we just stick them together, just like paper. And then we're going to put that artificial gene into the bacteria through many techniques like transformation, electroporation, and genomic integration. And the latest one is a CRISPR. The CRISPR, we can also use this one to edit the human genetic material. And through so many cloning process like this, then we have the strain that we think is going to be desirable for our production. And of course, we need to use this strain for the fermentation, and we do the analysis, and we do some processing to get the final product. like in this case, it's a PHA. So right now I'm gonna introduce to you, I'm gonna explain to you my latest research regarding the development of the strain PHB8. So I already applied for the patent for this strain and send some instance of this strain to the BCRC Taiwan so they can keep it. So as you can see that this is the pathway, the metabolism pathway of the PHP production. So it's quite complicated, I think. So the first thing in our lab, we already have the strain, which is designated as the N31, which is capable of using the crude glycerol, but it cannot produce anything. If you want them to produce anything, we need to add the gene that can help them to do the production. So that's why PHB1 as N31 with the additional FAR-CAB operand, which is right here the pathway, FAR-CAB. So once we added this one into the PHB, so it will start to produce PHB from 0 gram to 0.22 gram per liter in a flat scale. And then, if we take a close look at this pathway, we can see that this is acetyl-CoA, and that is a precursor for the PHB. But this acetyl-CoA also is a precursor to produce acetic acid. So you can see that in this graph, the green color column, that is acetic acid. They produce so many acetic acid. So that's why the second step to create the PHB8, which is a PHB2, is to delete this gene, PTA. So acetyl-CoA will no longer go to this pathway to produce acetic acid. So you can see that PHB2, the green part, acetic acid is very low already. And also the production is double, I think, 0.45 compared to 0.22. And for the next step, which is the PHB3, we add another FAR-CAB operand, and the final result is we have 1.4 gram per liter, which is 3 times comparing to the PHB2. And for the next step, so you see that acetyl-CoA also need to enter the TCA cycle to provide the energy for the bacteria. But we don't want a lot of acetyl-CoA to enter this cycle. Yeah, of course, we want some of them to enter the cycle. Otherwise, they cannot live. So we're just trying to reduce the expression of this gene, GLTA, which is to combine the acetyl-CoA and the oxaloacetic acid to the citrate. So in the end, we have just a little bit increase in the PHB production, 1.6 gram per liter. And then also for this step, for acetyl-CoA to the 3-hydroxybutyrate-CoA, which is catalyzed by the enzyme produced from the FAB. We have the NADPH here. And so that's why I enhance this PNTAB gene, so I have more NADPH. So the result, double the PHB production. And also, in order to have more acetyl-CoA, I also enhance the gene ACEEF to convert the pyruvate to the acetyl-CoA. So for this one, the production goes to 4.9 gram per liter. And this is so far in our lab is the highest performance for the PHB inside the flat scale. But if you want to scale up, we also try another strain, which is the PHB8. This is the final strain that we patent this strain, and we keep this strain in the BCRC. So we're adding another FAR-CAB operand, and this is the final result. Okay, this is my mistake, PHB6. The PHP6 fermentation only can yield 18.1 gram per liter. But for the PHP8, it's double, 36.3. And this is just the number when, at the time I write this paper. Right now, my assistant, he claimed that he can produce almost 50, 45 or something. It's very high. He fixed something in my feeding regime, and now he can produce so many PHP in 30 hours. So the productivity, 1.1 gram per liter per hour. This is actually very high for the production of PHP if we use the crude glycerol. Of course, if we use something like the sugar, it will be even higher than this number. But so as I just mentioned, our topic is about green chemistry. So I use crude glycerol, and through this process, I can reduce 97% of the glycerol out of the system, just doing the fermentation, and also get some PHP in the process. So that is my recent work. Of course, right now I'm also doing another work because I haven't published those yet. So my professor said that I shouldn't, expose it, right? So that is reasonable, right? But my future work for this one, the first thing is the PHP. Sorry. So the PHP is kind of very rigid. It's not flexible, and in order to apply it into many applications, we need the material that with a big range of characteristics. So that's why my next plan is to continue to develop the strain that can produce another kind of PHA, not just a PHP. So right now, what I'm doing right now is the PHP copolymer with the lactic acid. And another thing is to diversify the renewable feedstock sources. So our topic is about green chemistry, right? So we are supposed to have as many solutions as possible to utilize those waste to produce something valuable to our economy, right? So right now, we already developed the one that can consume crude glycerol effectively, and we already also have the strain that can consume something like the cellulose hydrolysate. So that is our next target. Maybe use those waste feedstock to produce added value products. And of course, not just bioplastic. We also want to expand our products type. So see the waste on our collection of E. coli, they can consume anything. They can consume so many kind of waste. And therefore, all we need to do is to study the pathway so they can produce something maybe even more valuable than bioplastic, maybe some biomedical products. I think that will be the very good future for this study. So that is everything for my presentation. Thank you for your attention.
Thank you for Dr. Xie, Michael. And then any question to Michael? Okay, please.
Yes.
Can you put your pathway on the screen again?
Yeah, I will show you the full, yeah.
You just said that your yield from glycerol to PHP is like 97% or something. But on this pathway, above the acetyl-CoA, that's pyruvate.
Yes.
From pyruvate to acetyl-CoA, you have to lose CO2. So you have to lose 49% of your molecular weight. How can you still possibly achieve a 97% of total yield?
First, I would like to thank you for your question, but I think there is some misunderstanding for this number of 97%. So actually, our yield, for 1 gram of crude glycerol, we can produce 0.3 gram of the PHP. And 1 to 0.3, right now, that yield is already very high, which is only 30%. But actually, this number, 97%, is the crude glycerol from the beginning I used. I put in, like for example, I put in 100 grams, but in the end, in the liquid culture, when I analyze, I only have 3 grams left. So that is the meaning of this 97%. But for, as you say, for the yield, yeah, the yield is only for 1 to 0.3, which is 30%.
So the yield is 30%. You
yes.
cannot say 97%. That's a misprintation.
Yes.
Waste reduction.
Yes, it is a waste
you
reduction.
lost 70%
yes.
to the CO2, so you cannot say that's a waste reduction. Glycerol is something very expensive right now.
Oh.
It's not a waste. It's a very expensive raw material, even more expensive than glucose right now. Okay, that's not a waste.
Yeah.
Okay.
Yeah.
And in fermentation, we present the yield.
Yeah.
How much material you put in, how much final product you got is 30%, which is correct.
Yes.
Okay. Another comment is you should only present what you know are the truth. You just said NatureWorks grows sugarcane and use sucrose, which is not true. NatureWorks production facility is in Blair, Nebraska, which I have personally visited. There's only corn. So they use corn starch, convert into glucose for fermentation. They're not using sugarcane. There's no sucrose involved in. Okay, this is a scientific presentation have to base on facts, not based on your imagination. Sorry for me to say that.
Thank you for your comment.
Oh, any question? Yeah, okay.
So what are some of the key challenges in implementing genetic engineering E. coli?
Sorry.
What are some of the key challenges in implementing genetic engineered E. coli solution on a larger scale?
For the larger scale.
Yeah.
So as Professor, sorry, I don't know your name, but you just give me the comment. So as he said that, like for example, from crude glycerol. So actually previously, when I checked the price from the time I doing this research, the crude glycerol is still the waste. But then from 2023, I see that the price for the crude glycerol start rise up. I'm not sure for what reason, but 2021, when the first time I started this study, the price I check on the Alibaba is just 150 less something per ton. But from 2023, I check, some people, they sell maybe 500, 600, even 800 US dollar per ton. So I think that could be the challenge. Because you see, 2 years ago, it's just a waste, but 2 years later, it's valuable product. We cannot know that. So that is the challenge. We need to utilize as many kind of the feedstock as possible. We cannot just rely on just one single feedstock, like the crude glycerol. And also, the professor also suggests me that 1 gram to 0.3 is actually very, very low yield. So yes, I already know that it's 1 to 0.3 is very low. So, but actually for the case of the crude glycerol, for my study, I try my best, just get 0.3. So that is also the challenge, because we use the waste, so it's not really desirable for the fermentation. And there's so many pathways that we haven't discovered yet. We just based on the thing that we already know to make this kind of product.
Okay, and then another question? Yeah.
是由这个赵永红赵教授哈，他是在 biotech 做的非常不错，实际上我们做了很多其他的领域。早期他们是用 glucose 是从这个 starch 啊，从 starch 或者是淀粉做 glucose 来做 lactic acid， 做 lactic acid。那现在后来呢，用这 biodiesel 有很多的这个 crude 这个 glycerin 就 glycerin 很便宜，所以我们就开始用这个 glycerin 来做 lactic acid， 这部分做的还不错，现在还继续在做。做完以后呢，就是 Michael 他负责就是用 glycerin 来做 PHP 就做 PHP， 主要是希望用 glycerin， 不要用这个 corn starch 跟这个或者用 sugar 跟这个所谓粮食来 compete。Food stock for corn starch, potato starch, etc. We are using just glycerin as a biodiesel, this very cheap one, to use this as a feedstock for the lactic acid and for the PHB, PHA like that to do it. The PHB and the PHA. 这个 PHB 是海洋生物 degradable 的 plastics， 这部分呢，直接是用 microorganism 用 E. coli 来制造它的这个所谓 polymers。直接不是 synthetic way， 完全是用 biotech way 来制造这个东西。但这部分呢，还是实际上他们还继续在 improve 当中。但是我们这个计划呢，现在是由科技部啊支持了2年计划，是由科技部 support， 所以我们视为呢 participate 来支持这个所谓产学合作。在这部分我们最重要，我们是视为公司做 biotech， 视为公司不是做 biotech 的公司，我们是做赶亚胶的，所以因为我进去啊，很想在这个训练一些 biotech 的人才。现在 biotech 人才是非常重要的人才，这很难。但是我们逢甲大学有这个基础，因为我有在逢甲这个关系，所以我们想一直支持在这个领域，所以我们花了好多的钱来支持我们赵云鹏赵教授来做这个 biotech 的这个合作。实际上，我们的意向意图呢，借重这个用 PLA lactic acid biotech 来做 lactic acid 或者做 PHP， 最重要是 training 我们这个研究人才，将来希望在其他领域来发展，过去 COVID-19 说 drugs 啊，像这个 antibody 啊，甚至抗衰老的这些 aconcynin、heparin 这种 这种所谓 medicine chemicals， 这个才有 value。实际上，在 lactic acid 现在竞争的很剧烈，在 PHA 是非常 cheap、非常 low end， 都是用大量的微生物细菌，就是它就可以做了。我们用 biotech 主要是训练人才，所以未来我们希望走到这种 specialty chemical， 甚至在其他领域来做发展。世卫公司只是投资，这就是 initial funding， 但我们最重要现在培养了3位这方面人才，是 postdoc， 还有这个博士学生啊等等这些人才啊。但不管怎么说，我们也是一个尝试。现在呢，有更多的年轻的人来学习这个 biotech。实际上，这个 biotech 这个人才在化学上是非常艰巨的，这面应该是要生物化学背景很强，所以啊，这是很辛苦的。一般人不是学这个 biochemistry， 你不太懂啊，要来做这个东西，看起来像是天书。实际上呢，这个问题我们还是继续努力。是不是我们世卫公司继续要支持这个计划？说实在话，因为这个是在公司来讲没有 commercial 的这个应用化是不愿意支持，所以我们也是赶快在这个领域，我跟赵教授也在谈呢。因为在逢甲有这么好的一个平台 biotech， 我们绿能中心也是做发酵，甚至在也有很多的这个人，他以前是在绿能中心，我们再转过来的，所以在这部分呢，我们是持续的来支持。怎么支持法呢？可能我们会有一些转变，因为公司是研究的是希望有产出，像我们做 academic 技术是没有产出的，所以在这一部分呢，应该是国家支持，所以我们希望在这方面呢，我们要做其他的这个变化。我们现在用 heparin 的来合成，用 biology 已经开始做准备，还有这个 aconcynin 这抗衰老的这些所谓纯粹的植物的氨基酸，这是非常重要的一些 就是 nutrient supplements 这块是从这个来仿生这个菇类里面这些产品，那这些呢可以创造出 value added 这部分就是他未来的工作。反正 biotech 是一个未来的科技啊，包括 这是疫苗啊，这是现在疫苗，还有我们现在吃的这个那个 drugs for COVID-19，这是非常重要的一些所谓这个 biochemicals 或者 drug chemicals 这部分呢，可能会创造很多的这个利益啊。那我想这块是我们大家继续努力。好，非常谢谢。不知道谁还有什么问题没有？呀，来。哎。我们是这个有大成改基因的，
你不是E. coli 的吗？E. coli 的。哦，OK，mutation，OK， 不是，我是说 mutation 很重要，他应该知道，就是说这个东西你要 controllable， 你一定要，对不对 ？How to avoid the mutation of the, because it's one of the most important
So, thank you, Professor, for your question. So, yes, you see that the mutation is actually occur from the DNA replication. So DNA replication, we have leading strand, we have lagging strand. The leading strand, there's almost no mistake, but the lagging strand have so many chance that you're gonna have the mistake to put that in there. So that is the reason that we can do the genetic modification, because of that lagging strand. But also in nature, that lagging strand from the DNA replication also caused a mutation, just like Professor mentioned. So I think, actually, based on my understanding, I think there's no way to stop the mutation. But I just suggest that we can have the solution to prevent the effect of the mutation to our production. So that's why, from the beginning, if we construct the strain, we need to keep what we call that the master stock. So from that strain, because based on my experience, if you use the E. coli, you stick it on the LB agar plate for many times, so the performance is just the same. So which means you can have one master lysate, which we know that the cell from that is 100% as our expectation. And then from that master stock, we can create many, we call that I'm not sure how to call that, but I would like to call that the working cell bank. Yes, thank you, the working cell bank. So we have so many working cell bank like that. We have 100. We may have, from the master bank, we can make 1,000 like that, and then we use that for production. So all of our production is the same as the performance of the working cell bank, and very close to the master cell bank. So that is, yeah.
Okay, we still have time. Okay, yeah, 5 minutes.
This is just $10 today.
Oh.
After 1 year, it might be $100, right?
Yeah.
So, will you consider this one as a valuable product or the waste? Because you just mentioned that waste reduction was 97, right? Yeah, it was right. Technically, scientifically, it was right. You cannot say if it is $100, it's a useful product. My point is, why it was not correct if the waste reduction was 97%. What was wrong in it?
So actually for that.
You said, you said that waste reduction was 97% was wrong. Why it was wrong?
So
if it is $100, it doesn't mean still it is a waste, right?
Yeah, actually, I think that 97 number is so misunderstanding for that 97. Yeah, I would like to yeah, just explain it, that 97. Yeah, so 97 is not all of the waste. I think the correct term should be 97% of the feedstock. I think it will be more accurate, 97% of the feedstock. It's not about the waste. Because actually in this stage of my study, I haven't have the chance or haven't have the equipment to check the CO2 from this kind of system. I just can check the crude glycerol and other intermediate product. So I just check as long as I have the intermediate product is very close to 0 gram per liter, then it means that the carbon source is, can say that is effectively consumed by the E. coli. Because I think to say that, yes, in the bigger picture, we need to consider all of the waste into the account, including all of the salt, the crude glycerol, the carbon dioxide, everything need to put into the account. But yes, that is my preliminary study. So at this stage, we just focus on to see if the bacteria can consume that crude glycerol or not. Or maybe in the future, if the strain can consume the carbon source from the feedstock that we want to add into the reactor.
Okay. Another short question and a short answer, okay?
Oh, from me? Okay. Okay, thank you. Sure.
Yeah, we still have 3 minutes.
Okay, sure. Okay, thank you so much, Michael, for your presentation. So it's really interesting for me, actually, because I'm working with this before, but now I'm like jumped to the biohydrogen production, but still, I'm really interested with this. So my question is like, the simple question is like, is it intracellular or extracellular?
What do you mean?
The production of the PHB.
The production is intracellular.
Intracellular. I mean, the product will be transferred outside the cell, or?
No, they are inside the cell.
Oh, maintained inside the cell.
Yeah.
So my next question is that possible if you like to use another host, or maybe inside the E. coli also possible to produce the PHB itself become extracellular? Is that possible to also to merge like the gene for the signal pathway to deliver the PHB itself outside the cell, so it make it easier to be extract from the cell? Is that possible?
So thank you for your question. So
Only 1 minute.
I never tried that before, but I saw somebody, they claim that they can do that. And we can also, but we also have another technology. I mean, not to let them to go outside, but using genetic engineering to destroy the cell. No need to take it out, just destroy them. Yes. Yes. So we let the cell to be. I'm not sure if I'm able to say this, but let's say that we have the way to break them.